Hello everybody, welcome to the first day of the BBE Summit 2021. My name is Darren Smith. I'm a Professor of Bacteriophage Biology at Northern University. I'm also the Research Theme Lead for the Microbial Environments. So we've got a really exciting day ahead associated with the speakers we have lined up to talk to you today. But firstly, what I'd like to do is go over a little bit of background regarding this research theme. And we know this is a mixed audience of biologists and non-biologists. So really what I'd like to set out is why we are looking at this microbial environments as a theme and what its role as part of the HBBE is, and what power and what it can offer to this community moving forward. So if we think about living in a microbiological world, we are completely surrounded by microbes. Every surface that we look at, every surface we touch, every skin surface, every organ in our body, mostly have some level of microbiota associated with it that have adapted and evolved to live in that environment. There's a lovely tree set out here by Spangler and Ettinger. It's a very simplified version of the microbial tree of life, but we can see there's differentiation between.
Aaron, your slides have not moved forward, so I don't know whether your slides are not advancing. We're just seeing we're seeing your PowerPoint interface with your. Yeah, that's worked. That's gone, but we're seeing your whole PowerPoint.
All right. Let's just undo my share and then see if we can push it to the actual screen itself, because it's showing on my second monitor. So I take it that'll be better. Is that now the full screen, Martin?
That is the full screen, and I can see your tree slide.
Perfect. Thank you very much. So as I was saying, there's a very simplified drawing of the tree of microbial life here by Spangler and Ettinger, and we can see the differentiation between the different phyla associated with bacteria, archaea, and eukarya. And if we think about if these are biologically active and grow, and if we think about overlaying this is the viral world as well. So each of the cell types associated with the tree are most likely predated by a virus of some kind. So when we talk about looking at microbial environments, they're multi-kingdom, and each of these environments are open to selective pressure. So even if we take this lovely schematic diagram, this cartoon by Peter Gamlin that was published in The Guardian, we can see that at each point along the kind of the GI tract, the stomach, into the colon of this cartoon, we see different bacteria. But this is a very simplified overview of that. If we resolve down to the tissue level, we would see different microbiota at different parts of this. So what we're trying to say is a number of studies go out and look into the microbial communities, but we're not really truly at the resolution of what's going on, what bacteria are there, what viruses are there, and then what metabolic activity is going on, what resolution do we need until we can start to understand what's going on. But these selective pressures can be associated with lots of different things. It's environmental, it could be to do with temperature, it could do with humidity. So if you think about that with respect to the built environment, then every room in your house likely has a different microbiota, most commonly shared probably with the inhabitants or animals within it, whether you open your window or not. But then if you go to individual corners of your room that are slightly cooler or warmer, then the likelihood is that you will see a different microbiota. We also need to know that if you remove a part of this microbiota, so if you remove a subset of this bacterial community, then the likelihood is that simple eukaryotes like yeasts and fungi tend to invade that space. So we see that in the gut, if somebody takes antibiotics, you see a reduction in diversity of the bacterial population, but then it opens a niche in that environment for the opportunistic microbes to jump in and take over. So diversity around us is I suppose it's a topical thing with the current pandemic, and we was trying to clean everything. And there's a massive trade-off now between sterility and this ability to promote a diverse population around us, because we are starting to ascertain that having a diverse population around us, or a certain diverse population around us, can promote health benefits. There was a study by Perka Karainen that was published in Nature Medicine that looked at, the development of asthma in kids and those children who grew up with a microbiota more like a rural environment that tended not to develop asthma. So there's this kind of, our immune systems are continually tweaked by these microbiota. So it's that trying to find out what that resolution we need to see, what are the positive microbes associated with our environment that we want to capture and enhance. So Gary presented the vision of the HBBE, and the last part is what falls into the remit of the Microbial Environments theme. So they modulate the microbiome to benefit human and ecological health. So the aims of the Microbial Environments team is mainly technology driven. It's to use the latest of omics technologies that we've been very lucky to be reach further support as part of this funding, moving from targeted to untargeted sequencing. So where you would look at a small part of a genome to look at differences and changes, to look at the untargeted sequencing. So you get a greater resolution of what's there. Within that, you can find genes of function. You can start to see other things that may be associated with shift. Maybe you've looked at gene expression of those genes, then you can start to get greater resolution of the function that's happening in that community at any one time. So that main focus that we are trying to employ as part of the team is this focus on multi-kingdom, the ability to overlay the different kingdoms. So you overlay bacteria, fungi, and viruses to get a greater resolution of what's happened in any one environment. Then if you can then longitudinally take that, so over a period of time, if you continue to take those samples, then you can start to look at what is causing those changes. I think we need to move to a metadata-rich community where we look at the environmental parameters that are supporting these samples that are being taken. And to do that allows us to start to use things like artificial intelligence to start to predict changes in the environment, and then the ability to augment or biostimulate that environment with a microbe of some kind in the future to come. But we wouldn't be we'd be remiss in our current strategy if we didn't focus on pathogen detection, especially viruses and sensors and sequencing. So when we look at the microbial themes, here is set out here in this kind of concentric rings of how all of the themes interact. Actually, I always see the microbial environment theme as always driving into the center of this, because it has good interactions with building metabolism, and their ability to look at how to use different enzymes associated with function, synthetic biologists that have been recruited, because if we're looking at untargeted metagenomes, it offers the ability to mine these data sources for novel enzymes. They also, with synthetic biology, there's a push for non-model organisms and clonal populations, but it may be that we can see some of this functionality within the environment that we can work with, and maybe that we can use this to support those studies. Again, with the living construction, most of the work is done with clonal populations, but over time, the ideal way to do it is to push this into mixed microbial communities. So if we do meet the Microbial Environments team, we have two Vice Chancellor Fellows, Dr. Angie Sherry and Dr. Matt Bastian. Angie is a microbial ecologist and molecular microbiologist. Her background's in geochemical processes associated with microbes in the environment. Her previous research is focused on degradation of oil, looking at organic matter and pollutants, and their aims within the HBBE is to decipher microbiomes of the built environment, to look at the complexity, very much so what we've been previously speaking about in the last slide. And she has a talk later on today entitled "The Translocation of the Microbiome on the Fungal Fiber Highways in the Natural and Built Environment," because she currently has projects that are funded by EBNet that do some amazing videos to be shown later on, and bacteria moving down fibers, which I think is always brilliant to see. And then Matt comes in to talk at 16:05, and we'll talk a little bit more about SARS-CoV-2 genomics in a moment, because during the pandemic, and because we are omics rich and we've been supported with extra equipment by researching, and we've utilized those technologies to support the sequencing consortium set up out of Cambridge as as a network of sequencing hubs across the north of England. So we're sequencing regionally in the north of England. And the theme lead, this is the only picture that is palatable, partly because it's half covered in black. But I'm a phage biologist. My kind of background is how phages alter microbial function after integrating into the chromosome. So if you see a bacteria that you think has a function, then all of a sudden you had a bacteriophage, then the function changes slightly. And then my pediatric Greg and Beatrice, whose PI is Angie, and Matt, whose PA is Matt Bashton, and they've also all got mirror board presentations at lunchtime today. This is the HBBE COVID response. As you can see, labelled in red are the guys who've helped support this, and Matt will give a bigger overview of how much we've done later in his talk, but currently we've sequenced over 20,000 viral genomes as a group. I mean, this is heavily supported by the kit offered by research team. Here's the guys just after Christmas, when the alpha variants were started. Everybody looks slightly tired if you look very closer into their eyes. So if we look at the agenda today, we've had the open, and then we have the great pleasure in introducing Jack, Richard, and Erica to present their work to us this morning. Then later on this afternoon, we'll get to see the VC fellows, but also Associate Professor Matt Unthank, who's looking at antiviral surfaces, and David Pearce, who are part of our home biome project associated with Dust that's NERC funded. Just to reiterate what Gary and Martin said, the mirror board poster presentations are at lunchtime, and the links can be found from the BBBE. And lastly, and the final thing is to just say enjoy the day, and please add questions for the speakers in the YouTube chat, and I'll be able to pass those on and facilitate those to the speaker at that time. So many thanks for your time. I hope you have a great day.